at Genentech. Welcome, everybody. So today, we're going to have approximately 50 minutes moderated discussion on precision prevention through the lens of genomics, and then that will be followed by a 10-minute Q&A session with our in-person and virtual audience. So, David, the field of cancer has really led in the implementation of genomics into precision medicine, both for diagnosis, but also for treatment. What opportunities are there to use genomics approaches for precision prevention?
Thank you, Ali, and thank you to the organisers for having me. As you rightly say, cancer has, for decades now, adopted an approach of targeting therapies to particular genomic mutations in cancer. You know, a cancer has a mutated genome that's, by definition, its problem, and drugs have been innovated over the last 30-odd years that can directly target actionable mutations to change the way cancer behaves and to treat it. So we've got a lot of potential there to think through how that could apply to prevention. Now, the challenge historically has been that the proportion of research spend in both academia and industry that's been dedicated to prevention research based on genomics is vanishingly small. Even if you characterize prevention quite charitably, you're still probably only talking about 5% maybe of the overall research spend, and that's across diseases, not just in cancer. So there's a problem there, but there's also a huge opportunity. As we increasingly get better at molecular characterization, not just of disease, but of human physiology, we're going to find more opportunities to intervene, not to treat disease, but to prevent, to maintain health. And that can be starting from using the genome to understand risk. And there was a fantastic example only 2 days ago where the UK regulator, the MHRA, for the first time approved a drug to be given in women at risk of breast cancer to prevent breast cancer. And that's a targeted agent to lower their breast cancer risk based on their family history, which is, of course, a sort of manifestation of genomic risk of disease. So the principle's there, it's been established, and we're excited to see where we can go.
Thank you, and thank you for organizing that for us a couple of days before this panel discussion. It took some
string pulling, but we did it.
So I really like the way you phrased this. I think we do need to start changing the narrative and talking about health rather than about disease as we move forward. Mark, I wanted to ask you, how can genomics and by extension multiomics, in addition to environmental factors, be integrated for the development of prevention approaches across the board?
Yeah, thanks. I mean, the way I think about it is that we need to bring together data in two spaces. We need to bring together information on individual genetic risk and lifestyle and other factors that play into disease based on population-level data to understand a little bit about the risk that an individual has of developing disease. On its own, that only gives you some index of predisposition. So we will need to combine that with other data that captures the progression from health to disease as individuals go through, and which will give a manifestation of whether that individual is progressing in line with the expected predisposition or different. So on the first hand, on the prediction, that's about genetics, it's about environment. It's an area that we probably feel most comfortable. We have most data, although there's an awful lot yet to be done in that space with regard to diversity, with regard to thinking not just about disease risk, but disease progression, and also thinking about the ways in which genetics and environment interact to influence individual risk. When it comes to the second, and thinking about how we monitor people as they track from health to disease, we're really just in the foothills of doing that at the moment, but we now have technologies with proteomics, for example, with metabolomics, thinking about PBMC, transcriptomics, cell-free DNA, cell-free RNA, that will allow us to get that kind of complex phenotyping that will allow us to track individuals. And that's going to be essential if we're going to identify people, not just people who are at high generic risk, but the people who are actually have moved out of health and are on their path to disease, because many of the therapies or other interventions that we're going to want to give for prevention are not ones that we can necessarily give to the whole population.
Thank you, Mark. I'd like to pick up on a couple of points there. You mentioned the need for large-scale, longitudinal, deeply phenotyped data sets that will allow us to identify those genetic risk factors and environmental risk factors with which we're more comfortable currently, but also molecular and other biomarkers of disease progression. What does the panel think, do we have the resources needed there, the data sets, the longitudinal cohorts needed to achieve that goal?
I think that's the big gap, to be honest. It's going to require a change in planning, not just around, we're all used to the fantastic amounts we've learned from UK Biobank and projects like that, which have certainly led the way, but to get to the longitudinal component, these things are going to have to be much more integrated into the healthcare systems, because that's, after all, the situation in which people are coming up and having their health monitored on a regular basis. And we need that both to collect the data, and we need that to be able to do the implementation once we've got findings that we need to implement. So I think that's the next challenge, is to find healthcare systems. The UK is in a good position, but there are others where we can actually achieve that congruence of both healthcare delivery and monitoring tied to the science that we need to do.
Thank you, Mark. Nicola, I wanted to ask you to weigh in on how precision prevention can, in fact, be implemented in healthcare systems. What is missing? What are the opportunities? What are the challenges?
So it's a really challenging question. I mean, all health systems around the world are under huge pressure right now. So logically, we should be going upstream and finding ways to intervene earlier. But if you look at health system budgets, it's incredibly difficult and risky for politicians to say, I'm going to take money from over here where it's needed and I'm going to move it upstream. And they ask questions like, well, which areas is the evidence strong in? And we've heard a little bit of that already from Mark and from David. And they say, well, if you were to do this, how would risk information really be integrated into clinical practice? Do we have clinicians who understand it? Do we have genetic counselors? They'd say, how would you engage the population when they have limited education, really, about how to understand such information, when they would essentially become a patient in waiting? And how would we manage that conversation? We haven't started having that conversation yet. And they would also say, when in an individual's life should this be collected? So in the UK, we've just started a really forward-thinking research trial, a newborn screening program with whole genome sequencing to really understand this. But I think that Mark is right. What we need are things like Our Future Health, implementation trials, which help us understand outcomes modeling, integration into clinical care, but also the health economics, so that we can really understand how those budgetary and implementation questions can be answered.
Thank you, Nicola. Nancy, you are an expert in linking electronic health records with genomic and multi-omic information. Do you think there are any systematic biases that have to be overcome in order to reduce structural health inequities in implementing precision prevention?
Precision prevention is a huge and hopefully attainable goal, but many parts of the world still have a lot of structural inequities in getting access to healthcare, especially high technology healthcare. But I think even within our existing healthcare system, genetics is a cause of some structural inequities. So when we talk about these biomarkers that help us understand how disease is developing in a person, how rapidly it is progressing, physicians think of these biomarkers as really dynamic measures of disease risk and progression. But 70% of the biomarkers, of the laboratory values physicians order on a monthly basis, are heritable. And a substantial fraction are very highly heritable. That means we are frequently misstaging people in terms of where they are in the disease process right now. We are doing harms because we didn't fully internalize, as these laboratory measures were developed, how heritable they are and how differential they are heritable across different populations in the world, and how the means and variances vary across world populations. So we have instantiated lots of health disparities because we haven't recognized those things. And I think genetics, bringing genetics into medicine, will allow us to use the genetics with the current laboratory values we have to do a better job in understanding who's really at risk of kidney failure, who's really having a bad reaction to their chemotherapies, and not just have normally low white cell counts. So I think it's a huge opportunity to use genetics to reduce existing structural health disparities as it comes into play for precision prevention and better diagnosis of disease.
Thank you, Nancy. You make a really important point about existing structural inequities in healthcare. But Segun, I wanted to ask you about potential future structural inequities and access to the promise of genomic medicine.
Yeah, that's a great question. So genomic itself is great, but currently you see that genomic there's imbalance in genomics. About more than 86% of genomics are being focusing on just one population. Africa, for example, there's only about 1.1% of genomics that have been done in African population. So what that means is that if we have all the best technology and we can prevent, we can even predict, it means that will be focusing on a population that represents about 16% of the whole world. So we can do better. So if genomics if you can study all population, including those indigenous population in different countries, Canada, Australia, and all over the world, and also African population. So there's need for genomics is very powerful. It can be used for prevention, like we've all mentioned today. It would be great to use genomics for prediction. Imagine if we can use the power of genomics to identify a subgroup of people that are going to have some cancer in the next 5 years or 10 years. That would be great, but that has to be done for all populations.
Thank you, Segun.
Yeah.
I think in the future, we might envisage everybody having an app on their phone with their genetic risk or polygenic risk, informing of risk of disease and therefore also relevant interventions. But does everybody want to have that information? I wonder what the public's perception of genomics in prevention would be and how that differs across different countries.
No, but let's not over-index on the genomics. I know genomics is important, that's why we're here today, but a lot of the differences between individuals relate to non-genetic factors, income, and so on. We've heard about, I've just moved to the States 5 years ago and getting familiar with the US health system, and you see so much disparity in that. For example, black women with breast cancer have much worse outcomes in the States. Now, part of that probably is genomics, but a lot of that is to do with income and access to inequitable access to healthcare. So we shouldn't over-index on the genetics. We know that the polygenic risk scores that we've got are quite good, but they're not fantastic predictors of disease risk, and that's why we need to fold in the non-genetic factors. And that's why, going back to my earlier question, we also need to fold in longitudinal measures so that we can track people. Not everybody with the same polygenic risk score and not everybody with the same environmental circumstances is going to develop the same diseases at exactly the same time. People are going to, for various reasons we don't understand or through stochastic factors, are going to progress at different rates. And it's that integration that we need, to be honest. And people don't need to see just their polygenic scores in isolation, I would say.
No. David?
So I completely agree with Mark about the need to integrate genomic risk with wider intrinsic and extrinsic factors that also confer risk. You asked, Ellie, whether people will want to know and then be able to act upon it. So I work for a charity it's a research funding charity in the UK, and we do a tremendous amount of patient and public engagement to try and ask exactly those questions. What we see in cancer is that there are several very highly penetrant genomic risk syndromes in cancer. So inherited risk that's very high that confers, a pretty intense risk of you developing cancer. And people with those risk syndromes are very, very engaged with the concept of risk, with the concept of risk driving their behaviours and what they will and won't accept in terms of medicalisation. And these are healthy people, but who've inherited risk. And they want to know, and they want to be able to act on it, and to keep themselves healthy. Now, there's a continuum there, as Mark indicates, from, I've inherited a really high-risk gene to I have a profile of risk across many genes that puts me a little bit at risk. And then, the motivation to then adhere to healthy lifestyle is different. But generally, when we engage with the public and ask them, would you want to know, if I had the sort of magical doodad that can tell you your lifetime risk of X, Y, and Z, generally people say yes. The challenge is it actionable? That's what they want. They want knowledge in order to inform decision-making that they can lower their risk.
Thank you, Nancy.
I just add to that there have been a lot of studies on this. It's a complicated thing because when you first ask people, they'll always say they want to know. If you go deeper into how actionable the information will be for them, when there's nothing they can do to modify their risk, they're somewhat less interested. But they remain interested if it's something that they would do something financial about. If it's high risk of a late onset disease where they might make decisions in terms of the way they do their late life finances, they're still very interested in learning that information. So there's nuances to the way you ask the question of them. Yeah. In terms of what people actually want. But I also wanted to really reinforce what Mark said about genetics is one of the most important ways we can learn about the non-genetic risk factors. The more genetic information we have, the clearer the non-genetic risk factors for disease will be. And while astounding advances have been made in our ability to modify genomes to reduce risk of disease, it will always be true that modifying the environmental risk factors for disease will be much less expensive and much more accessible to people. And so learning the biology of the non-genetic risk factors may also inform us on new drugs that we could use to reduce risk of disease. It's a huge opportunity now as more and more genetics comes into medicine to really focus in on non-genetic factors that can be environmental exposures, but also some kinds of social determinants of health that actually modify long-term biology. So it's a huge opportunity for us to improve health by modifying the things that we can modify most easily.
So we've had some very interesting experience with consent at Genomics England, because obviously we ran the 100,000 Genome Project, and now that's run into providing a live service of whole genome sequencing of rare disease and cancer patients. And one lesson we've learned is you have to not only have a proper consent process, but you also have to have the right people available to go through that consent with the right amount of time, which means you have to have enough genetic counselors available within your health system if you're going to provide genomic sequencing or genomic testing. But secondly, that it is not just about having the test for yourself. We have a 98% consent rate to participation in research. So many of those who have whole genome sequencing for rare disease and other conditions actually do not return a result of a diagnosis to them, but they all articulate, or the vast majority articulate, that they want to participate in research programs that we run in partnership with industry and researchers in order to improve diagnostics and therapeutics for others like them in the UK and around the world. So that's a really important part of the dynamic, which I think is significant to understand in designing these programs. The second element, I just want to come back to Mark's point about genetic data and genomic data not being everything. It's hugely important, but it has to be wrapped in structurally with all the other modalities. And we're running a program called Cancer 2.0, and we can only do this because of the advances in technology. We have about 22 petabytes of data at the moment, and with the Newborns program, we're going to double that. Without cloud, without the advances, there is no way we could host that data. One genome is the equivalent of the hard drive of a current laptop, right? And so every time we sequence, that data just explodes. But what we can now do is we can add to that pathology data, which is being digitized. We can add to that imaging data, which is being digitized. And this means that you can bring together all those analytics, which clinicians had previously been looking at in silos to try and understand what was going on with clinicians. And this will lead to new insights and accelerating diagnostics, which is really exciting. And then if you bring in what we have in the UK, which is our longitudinal data for clinical understanding, whether it's cancer treatments or GP history, then you have something which is incredibly powerful, which we just technically couldn't have done until now. So this is an exciting moment, and we're just at the beginning of it.
Okay, I can talk about the experience that we had in Uganda, where we do some genomic study. So at the beginning, we test people for various kinds of disease, including the cholesterol. And you see people showing interest in knowing what is going on in their life. So they want to know whether they have. In our setting, it's even worse because in one study, it was about 100% of the population that have high cholesterol are not even aware that they do have that. So what is very important is that there is a lot of engagement between we, the scientists, and also the people that we are the participants that we are working with them. So we have to see them, we have to build trust with them. If you remember, during the COVID, some people are not going to show interest in vaccines, some are going to show interest in vaccine. You don't blame anybody for that. So they need to trust us if you are going to provide something for the public. And I think it's the same thing also with the genomics. If people are going to trust what we do, there has to be that element of trust between us and them. They have to see the reason why what we are doing is very, very important for them, and it will help them to have a better health.
Yeah.
I just wanted to sort of layer on something to some of the concepts that Mark and Nicola just touched on, of looking at sort of multimodal data from different sources and that trajectory over time, over an individual's or patient's life. So in cancer, there's been a term coined called interception. So you've got a trajectory and a continuum between complete health, somewhat at risk. There's a few cells have started misbehaving, or there's some slight aberrations, but they're not really disease. They're just a bit wonky. And then into sort of full-blown disease, but occult disease, where something's gone wrong. You've got early disease, but you don't feel it. You've no symptoms. And then a journey into kind of clinically frank disease, where symptoms become apparent. And that's a continuum. There's no sort of, in most non-infectious diseases at least, there's no black and white, yes and no. So that proceeds over time. So if you could harness, your health records across, all the touch points that you've had with clinical service over your life, every time you went to your family doctor, every time you had a scan, every time you had a blood test, there's a tremendous amount of longitudinal information there about trajectory. And if you could then, bring in the genomics onto that, if you're sequencing every newborn baby, and then suddenly you've got this wealth of information, you've got the opportunity to intercept, which is to, target those very early changes before it's become established disease and intercept an aberrant process. So there, you don't have to give everybody who's at somewhat risk a drug, but you could target it to people who have started a journey to deviation away from health towards disease and intercept at that point. And that's something that I think is really exciting. It's where we're sort of focusing a lot of our efforts.
And then another point to pick up on that, which I think also Nancy addresses, is that longitudinal record allows you to be assessed against your historical self, not against population norms. Yes. You know, the cyclists in the Tour de France, when they're done for doping, it's not necessarily because they got a testosterone level that was outside the normal range, but it's gone from 6 to 12 since last year. It may well be within the normal range. So it's that ability to understand where was your homeostatic happy norm and look for departures from that, which you'll probably pick up well before, in many circumstances, it becomes an abnormal lab value.
But if we could cycle back just briefly to the question you asked me first, which is how do you get these sorts of precision prevention policies into public health? One of the big barriers, I think, to getting prevention into public health is that they've been one-size-fits-all. And people do not like one-size-fits-all prevention policies. It's don't smoke, don't drink, don't have fun, don't eat cake. And actually, if you're given evidence that you as an individual have a specific risk and there is something that you can do about it, that is much more engaging and convincing. And I think that has much more policy grip than what we've been trying to implement until this point.
Absolutely. So one thing we have, I think, all talked about are data, big data, hard disks and hard disks worth of data. Do we have the tools needed to integrate all of that deep and longitudinal information to build those prediction models that bring together environmental risk factors, genetic, multi-omic biomarkers, social?
Yes.
Brilliant.
If you were around this morning in the sessions, there's a huge array of incredibly exciting applications of these technologies. But I feel like the tools, the computational tools, have outpaced the science we're doing with them. So I think the tools are ready, and I think we're just figuring out how best to use them. But the potential is vast.
I think to one of Nicola's points also is that we need to do a better job of federating large-scale data that are being collected in many medical centers now so that we take full advantage of all of the data to learn as much as we can, as fast as we can. I think the computational environments and the tools are ready, but it's hard to access all of the data that is actually useful in the ways that would be the greatest good for the greatest number. And I think it's possible to do that. We just need to work with the lawyers who control medical centers to make sure that all the privacy is protected. But there's so much more we could be doing if we could put all of these data together.
Yeah, I agree, Nancy. We need to collaborate more. So we need to understand that we are trying to solve a global problem. And the research should not be one-sided. So we have this technology, but is it accessible by everyone all over the world? I remember particularly when I was doing my master's degree in Nigeria at that time, I was running a program that was running on my personal computer for 3 months. So I came to Heidelberg here in Germany, do internship. That same program ran for about 3 seconds. Just look at the difference. So this technology, we have them, but they are just being in one place, in the Western country. So this is a global, we're talking about global heads. Everybody needs to have access to what we are talking. That's why I agree with you about the federated technology, where data can be put in one place and everybody can access it from where it is without any barrier.
Yeah, I mean, you're right. And of course, we need complex health and other data to come together. But that is very personal and sensitive data. So at the same time, We hear, we have these currents of increasing data privacy of countries regarding their genomes as national sovereignty, partly because of historical abuses of that. So we have to work out how do we manage those two competing problems. So it's maybe not so much about data being shared. We should share data where we can, but about knowledge being shared. So I'm excited by these opportunities around federated learning and so on in the AML space, because we are going to need massive data sets. Yep. We want those data sets to be representative of entire populations. Otherwise, we're going to build models just on wealthy Europeans with very limited genomic and environmental diversity and expect them to work in other parts of the world, and they won't. So I think we have to have imaginative solutions that allow us to share the learnings, because sharing the data is going to be very difficult in many circumstances for perfectly legitimate reasons.
Thank you. David?
So we've talked a lot about understanding risk and then using genomics to kind of give you the population to understand the mechanisms of that risk and where you might intervene. We haven't actually talked so much about how then you develop the preventive interventions themselves. And Mark, if you can put your fingers in your ears, I'm just going to bash the pharmaceutical industry for a minute. The pharma industry has a model where they treat disease. Of course they do. That's what they're there to do. But developing preventive agents has not, by and large, been on their radar. We've got kind of one exemplar really in statins for a kind of global, blockbuster treatment that's massively reduced the risk of cardiovascular disease. It's prevented, untold millions probably of cardiac deaths. But beyond that, there's not many great examples of industry having really gone for it. So there's a real kind of research and development challenge there. There's also, I suppose, a political and regulatory and incentivization challenge. Why should pharma go out their way to spend? And it would take billions, to develop a new preventive drug. The trials that you have to do are immense, because you imagine, you're taking a population of healthy people and treating a bunch of them with a drug and then waiting for the people who didn't get the drug to get disease. These are big, big studies. So there's a lot of upfront cost, dependent on a hope that the system will then reimburse them appropriately for that cost if they get this preventive drug. And that, I think, is a huge unknown, right? So there's a massive kind of health economic challenge here as well.
I think you've answered your question why pharma doesn't go there. I mean, the regulatory aspects are really difficult. But that's where this stratification of prevention, so that you can identify, for example, individuals who are ostensibly healthy but are fast progressors on a trajectory to disease, that allows you to think about the kind of trials you could do that would allow you, within a reasonable time, and cost to investigate those. So pharma's interested in treating as many people as possible and bring their medicines to as many people as possible to execute the biggest benefits. So there are structural challenges here that need to be overcome, I think.
Thank you. I am told that we have reached the end of the discussion part of this panel, and now I'll hand over to Adam for Q&A.
So just a reminder, our online audience, please drop your questions in the chat. And for our physical audience, please raise your hand if you have a question. So while we're getting a mic over to this person just here, let me ask a question from the online audience first. So risk is a difficult concept for all of us, I suppose, to understand, but maybe especially when it concerns our own health. How do we help patients understand what it means for their future actions if they have a 5% risk of this, a 10% risk of that?
I mean, that's a huge challenge. And it's not a challenge that scientists are best placed to answer more in most cases. So it's a communications issue, and I agree with the online questioner. Humans are terrible at assessing risk by and large. We kind of walk around petrified of minute risks that never are really going to hurt us, and then we blatantly ignore huge risks when we're driving or crossing the road or whatever. So it's a communications thing, and it's understanding, as we discussed earlier, what people want to know, and then finding the best ways to communicate what they need to know. which is it's a behavioural science challenge.
Yeah, I'm often struck by the different ways that society treats health information and financial information. We give people a lot of leniency to make decisions about their pensions, their investments, their mortgages, and so on. And we have certain legal tramlines that we use to try and dissuade bad actors for getting involved. But those have huge impacts on people's well-being and the well-being of their relatives in the same way as genomic and other health. So I don't have any particular lessons from that, but I think we can sometimes take some thoughts from the way that we treat those other types of data and decision-making within people's lives.
I think also, I think also there's some risk in which there's genuinely no actionability. And that is very difficult discussions to have, and there are trained clinicians within the health system who are accustomed to having those discussions, and those are very difficult processes to go through. But a lot of risk in cardiovascular disease or even in cancer does have interventions which are possible, whether that's screening or lifestyle changes or steps that can be taken, which involve check-ins and and also, of course, lab results, which can observe the process and mean that you can check risk and you can change it. Type 2 diabetes is an obvious example of this, where we routinely have risk assessment, and you can reverse type 2 diabetes by changes of lifestyle and interventions medically. So we do have this within our health system now, just in a very small way.
I would make the same point. Patients are more and more used to getting information about risk factors that they have. LDL cholesterol in cardiovascular disease is a great example. They understand if their LDL is high, they're at increased risk for myocardial infarctions and other bad outcomes, and they can be put on a statin and lower their risk. They can stop smoking if they're worried about their lung cancer risk. So I think patients have started to internalize a lot of these risk concepts. We have a long way to go to be able to convey the complexity of risks that we're going to be able to access with genetics. But we have a lot we can build on already.
So I think we can do better the way we communicate with patients. Patients understand that if they are going to invest their money in a company, and they are told that if you invest £100,000, this is your risk. They understand that. And they understand the risk involved in insurance. So why should it be difficult for patients to understand the risks about their health? The reason is because we don't communicate that correctly to them.
Yeah. There's another sort of lens on this, which is about diagnosis. Do you want to be undiagnosed, even if you're asymptomatic? So I tell a story sometimes, but I will today. I was 30 years undiagnosed with a rare disease and very symptomatic, in and out of hospital and so on, and was diagnosed accidentally. And I can tell you it's no fun being an undiagnosed patient within a health system. You bounce around, you have lots of inappropriate interventions, the wrong drugs, it makes you sicker, people look at you like it's all in your head, they say it's terrible. About 30% of rare disease children die before their first birthday. And so the majority of patients would prefer a diagnosis because it leads to a better experience. But it is very personal, and there will be some people who will say, I don't want to know. So that's what consent is about, and that's what having the discussion with your clinician is about.
Thank you. We have a question from an audience member just over here. Would you be able to stand up for your question? Thank you.
Hi. Sorry. So thank you very much for a really amazing talk. It's really a topic that is important for me. So I have perhaps a naive question which follows one of the discussions that David, you mentioned, is that, okay, I can imagine a situation where we have, really precise medicine at the genomic level, where we can predict, that gene A is connected to this subtype of disease which a patient has, and gene B, C, D, all of this situation. But then there could be a situation where, we know that the gene is potentially connected to that disease, And we know that there's potentially a small molecule or a therapy that can target that. But we've never tested that because it's predicted based on genomics. And I can imagine that that can be the case for many, many people. And I can also imagine that, for example, it'll be hard to get trials or to get robust data on that because the medicine is so precise. There is very little people with that problem. Do you think that is actually a concern, or do you think that's just... sorry.
Thank you.
Yeah. So that's, it's a precision medicine question, isn't it? That the more that you stratify down a group and the more precisely you can dissect apart the molecular mechanisms of disease, the more your strata will become vanishingly small. And so, and it might just be, it won't necessarily be a single gene thing. It could be, a polygenic thing plus all your different exposures. And then suddenly it's only you that's got that unique mix of stuff. And so maybe you'd benefit from that drug, maybe not, but we can't do the trial because it's N of 1. So yeah, I'm just describing what you just said in more words without offering any solution whatsoever. So I think we need to evolve the way that we do research, but to account for exactly that problem. I think that the days of very large scale, very simple trials are probably numbered. And that we're going to need to find a more sophisticated way to develop interventions, particularly in very targeted, prevention settings, as you're articulating, that account for that diminishing sort of population and increased molecular stratification. The same old system won't work.
Nancy?
Things are already happening, though, going in the opposite direction. A drug company identifies a gene that they think is a good target for a certain rare disease. They've got the drug. Now they need help finding all the patients who have that condition. And I think that's where, some of the AI and machine learning approaches with full-scale electronic health records are extremely helpful because you can use the very, very, very early stages of disease, patterns of biomarkers and lab values to precisely identify the patients who then would really benefit from this drug in the context of a quite rare disease. There will be some true N of 1, but honestly, there are always more people than you think affected with any rare condition.
Yeah, amen.
Yeah, and to add to that, we design drugs to interact with a particular target, but they affect pathways and they affect pathophysiological processes that may well go beyond that. So that's another area that, again, I think AI/ML is going to help considerably as we move within drug development to doing large-scale studies on appropriate cellular and organoid models where we perturb both the genetic and the chemical space at the same time and get to a more fundamental molecular and pathophysiological dissection of disease. I think that's going to make some of these potentials to expand beyond a very limited, what may appear at first to be a fairly limited indication space and have much wider impact.
We're running out of time for the day, but I saw a question back here, and I'd be remiss if I didn't try and sneak in one last question and answer, but please keep it brief, both in the question and your answers.
Yes. Just based on today's technology, because we're doing computational reassembly from fragments, there are always a lot of errors in what we consider to be gold standard data. If you then go compare it to long read sequence data from PacBio, how do you see that going forward? Because it may be that a lot of the data we have turns out to have a lot of errors in an underlying when we can in the future go to longer read sequencing.
It's for you, Mark.
But I think in general, a lot of legacy data which wasn't collected for this kind of purpose is not going to be terrifically useful. But with the falling cost of acquiring data, and if we think about acquiring data in ways that make them more appropriate for future wider use with AI/ML and for sharing and some of the things we've discussed before, then I think we'll solve some of those problems. Within our company, we're trying to avoid spending a huge amount of time trying to curate legacy data unless it's incredibly valuable, because to be honest, we can generally rederive it at much lower cost and with much higher resolution and much higher accuracy now than we could 2 or 3 years ago.
Thank you. How fitting that the last question should be about emerging new technologies. I think at the panel today we've really captured the essence of precision prevention and the power of genomics, and how, in congruence with emerging enabling technologies, there's huge promise going forward. So with that, we will close the final plenary table discussion for the day. Thank you all for joining us today at the Falling Walls Circle. And we'd like to invite you to join us at the evening reception at the Art and Science Forum at Holzmarkt Zelchen. Just follow the red balloon out of here. Thank you.